Obesity-induced insulin resistance represents one of the most critical and escalating global health challenges of the 21st century. It acts as the primary pathophysiological driver behind a cluster of dangerous metabolic disorders, most notably type 2 diabetes mellitus, non-alcoholic fatty liver disease, and cardiovascular disease. According to data from the World Health Organization, an elevated body mass index, or BMI, is a major independent risk factor for non-communicable diseases and is directly responsible for approximately 5 million deaths globally each year. Understanding the deep, intricate scientific reality of how excess adipose tissue destroys metabolic health is absolutely vital to addressing this global epidemic. A definitive narrative review published in the journal Molecular Biology reports, titled Insulin Resistance Induced by Obesity: Mechanisms, Metabolic Implications, and Therapeutic Approaches, maps out the precise cellular pathways linking weight gain to metabolic failure, analyzes the systemic consequences across vital organs, and details the medical breakthroughs being deployed to counter this condition. Insulin then travels to target metabolic tissues, such as skeletal muscle, adipose, and liver tissue, where it binds specifically to the extracellular alpha subunit of the insulin receptor on the cell surface. This binding event induces an immediate conformational change that activates the intracellular tyrosine kinase domain of the receptor's beta subunit, causing it to undergo self-phosphorylation. Once activated, this receptor triggers the recruitment and tyrosine phosphorylation of a key docking protein known as insulin receptor substrate 1, or IRS1. The fully activated IRS1 protein then serves as a platform to recruit an essential lipid kinase enzyme called phosphatidylinositol 3 kinase, commonly abbreviated as PI3K. This enzyme catalyzes the conversion of a membrane phospholipid called PIP2 into PIP3. This newly formed PP3 acts as a crucial molecular beacon at the inner cell membrane, recruiting phosphoinositide-dependent kinase 1, or PDK1, alongside a protein complex called mammalian target of rapamycin complex 2, or mTORC2. Together, these complexes phosphorylate and activate protein kinase B, which is widely known as ACT. Activated ACT is the master molecular switch for downstream glucose transport. It triggers intracellular storage vesicles holding glucose transporter 4, or GLUT4, to rapidly move toward and fuse with the plasma membrane. This mechanism physically opens the cellular gates, allowing glucose to rapidly flood out of the bloodstream and into the cell to be utilized for metabolic energy. Simultaneously, ACT phosphorylates and disables an enzyme called glycogen synthase kinase 3, or GSK3. Disabling GSK3 frees up another enzyme called glycogen synthase, which allows the liver and muscles to safely convert excess glucose into stored glycogen for future energy needs. However, in an obese state, this highly organized, elegant molecular pathway is systematically dismantled by 3 interconnected destructive forces: adipose tissue dysfunction, chronic low-grade systemic inflammation, and intracellular oxidative stress. The first destructive phase begins within the expanding fat tissue depots. Adipose tissue is biologically designed to act as a safe, flexible buffer for excess nutrients, storing energy in the form of neutral triglycerides while protecting other vital organs from lipid overexposure. But in progressive obesity, this tissue reaches its absolute functional storage threshold. The individual fat cells undergo severe adipocyte hypertrophy, meaning they stretch and enlarge beyond their physiological limits. As these individual cells outgrow their local microvascular blood supply, they experience profound cellular hypoxia, or a lack of oxygen. This hypoxic, hyperstretched state causes severe endoplasmic reticulum stress and triggers massive, uncontrolled lipolysis, which is the chemical breakdown of stored fats. Because these dysfunctional, hypoxic fat cells can no longer properly store lipids, a massive wave of non-esterified free fatty acids overflows directly into the systemic circulation. These free fatty acids travel through the bloodstream to non-adipose organs that are entirely unequipped to handle fat storage, such as the liver, skeletal muscle, heart, and pancreas. This misplaced fat accumulation is called ectopic lipid deposition, and it causes severe cellular poisoning known as lipotoxicity. Inside the liver and skeletal muscle cells, these excess fatty acids are converted into highly toxic lipid intermediates, specifically diacylglycerols and ceramides. These toxic lipids activate destructive cellular stress enzymes, most notably protein kinase C. These stress enzymes physically block the normal insulin pathway by causing abnormal serine phosphorylation on IRS1. This effectively cuts the communication wire between the insulin receptor and the downstream PI3K pathway. As a direct result, even though the pancreas is producing a vast surplus of insulin, the cellular gates remain completely locked and glucose is barred from entering the cells. The second destructive force is chronic low-grade inflammation. The hypoxic, stressed, hypertrophic fat cells begin to release molecular alarm signals in the form of pro-inflammatory cytokines, including tumor necrosis factor alpha, or TNF-alpha, and interleukin-6. These chemical alarms cause a massive infiltration of circulating immune cells into the adipose tissue. Critically, the local macrophages undergo a polarization shift, changing from a helpful, anti-inflammatory M2 phenotype into a highly aggressive, pro-inflammatory M1 phenotype. These M1 macrophages cluster tightly around dying fat cells, forming macroscopic structures known as crown-like structures, and pump out a continuous, highly concentrated stream of inflammatory signals. These circulating cytokines travel throughout the entire body, activating master inflammatory pathways in the liver, muscles, and pancreas, specifically the IκB kinase beta, or IKK beta pathway, and the c-Jun N-terminal kinase, or JNK pathway. Furthermore, the liver and pancreas activate suppressor of cytokine signaling, or SOCS proteins. Together, JNK, IKK beta, and SOCS proteins act as molecular saboteurs. They physically bind to IRS1, targeting it for chemical destruction and preventing it from interacting with the activated insulin receptor. This completely halts the downstream activation of ACT and stops the translocation of GLUT4 transporters to the cell surface, locking the blood sugar entirely outside the cell. The third destructive force is oxidative stress, caused directly by mitochondrial dysfunction. When metabolic cells are constantly flooded with an overwhelming supply of excess glucose and free fatty acids, the mitochondria are forced to work at maximum capacity to oxidize this fuel. This constant, high-velocity metabolic burning eventually damages the delicate internal electron transport chain of the mitochondria. Instead of cleanly creating adenosine triphosphate for energy, the broken mitochondria begin leaking highly reactive, unstable oxygen molecules called reactive oxygen species, or ROS. This sudden surge in intracellular ROS damages cellular lipids, proteins, and DNA. Crucially, high levels of oxidative stress further accelerate the activation of the destructive JNK and IKK beta inflammatory pathways. This creates a vicious, self-sustaining pathological loop where fat accumulation causes inflammation, inflammation causes oxidative stress, and oxidative stress worsens insulin resistance. The systemic metabolic implications of this three-pronged cellular destruction are vast and debilitating. In the liver, insulin resistance impairs the organ's ability to sense circulating insulin, meaning it fails to suppress gluconeogenesis and glycogenolysis. As a result, the liver continuously pumps excess glucose into the blood even when blood sugar is already dangerously high, leading to fasting hyperglycemia. Concurrently, altered lipid metabolism increases de novo lipogenesis, packing the liver cells with triglycerides and driving the development of metabolic dysfunction-associated steatotic liver disease. In skeletal muscle, which is normally responsible for over 80% of post-meal glucose disposal, the failure of GLUT4 translocation drastically reduces the body's overall glucose clearance capacity, causing prolonged postprandial blood sugar spikes. Meanwhile, the pancreatic beta cells initially attempt to compensate for this systemic resistance by undergoing hyperplasia and hypersecreting massive amounts of insulin. However, over time, the combination of chronic glucotoxicity, lipotoxicity, and local inflammation causes pancreatic beta cell exhaustion and apoptosis, leading to a permanent decline in insulin production and marking the official transition into clinical type 2 diabetes mellitus. To combat this multifaceted cellular destruction, modern clinical medicine has established several therapeutic tiers designed to intervene at specific, targeted points within these dysfunctional pathways. The first tier involves classic, time-tested insulin-sensitizing medications. Metformin remains the foundational first-line therapy worldwide, working primarily by activating adenosine monophosphate activated protein kinase, or AMPK, in the liver. This activation directly suppresses hepatic gluconeogenesis and improves peripheral insulin sensitivity in skeletal muscle tissue. Another class of sensitizers, the thiazolidinediones, act as powerful agonists for the nuclear receptor peroxisome proliferator activated receptor gamma, or PPR gamma. These drugs remodel adipose architecture, promoting the creation of small, healthy, insulin-sensitive adipocytes, and helping to redistribute toxic ectopic fat away from the liver and muscles, safely moving it back into subcutaneous fat depots to reduce systemic lipotoxicity. The second tier utilizes modern metabolic innovations, GLP-1 receptor agonists and SGLT2 inhibitors. GLP-1 receptor agonists, such as semaglutide, mimic natural incretin hormones to significantly enhance glucose-dependent insulin secretion from the pancreas, delay gastric emptying, and act directly on the central nervous system to suppress appetite. This leads to profound weight loss that resolves adipocyte hypertrophy and tissue hypoxia at the root source. Meanwhile, SGLT2 inhibitors, like empagliflozin, work through an insulin-independent mechanism in the kidneys, blocking glucose reabsorption in the proximal renal tubules and forcing the body to excrete excess sugar directly into the urine, rapidly reducing systemic glucotoxicity. The definitive conclusion of this extensive scientific literature review highlights both a warning and a profound shift in medical perspective. The researchers conclude that obesity-induced insulin resistance cannot be viewed merely as a passive consequence of weight gain, but must be treated as a highly active, complex, and dangerous web of cellular signaling crosstalk. It is a condition where adipose tissue dysfunction, macrophage-driven chronic inflammation, lipotoxic ectopic fat accumulation, and mitochondrial oxidative stress reinforce one another in a destructive cycle that ultimately causes systemic metabolic collapse. However, the authors emphasize that understanding these exact molecular pathways, researchers are currently investigating direct anti-inflammatory therapies, using specific monoclonal antibodies and small molecule inhibitors to block TNF-alpha, interleukin-6, and the IKK-beta pathway directly, thereby preventing immune-mediated destruction of the insulin receptor link. Concurrently, the medical field is shifting toward personalized medicine using pharmacogenomics to analyze an individual's unique genetic code. By identifying specific variations in a patient's insulin signaling genes, doctors will soon be able to predict exactly which medication combination will work best for their specific cellular profile. Ultimately, the document concludes that by shifting away from a one-size-fits-all model and embracing multi-targeted, molecularly precise therapies alongside early lifestyle interventions, modern medicine can finally break the vicious cycle of obesity-induced metabolic failure, transforming our approach to insulin resistance from a state of chronic management into a highly treatable, mathematically precise science.